last session we have completed androecium, the member stamen, parts of stamen, structure of young anther, structure of mature anther, steps involved in microsporogenesis and structure of mature pollen grain or microspore. It is also known as male gametophyte. In today's session, let me start with the female reproductive whorl in flower. It is known as gynoecium. The members of gynoecium are called carpels. They are also known as pistil. Each carpel has three parts. The upper portion, which receives pollen grains during pollination, known as stigma. The tubular portion is style, through which pollen grain germinate, pollen tube penetrates to carry the generative cell. And the most fertile important part of carpel is ovary. Now this ovary is a site of megasporogenesis. It is a site for double fertilization. It is a site for embryonic development, and it is a site where the embryos are protected within the seeds, and this ovary develops into a fruit. Let us study one by one. Consider parts of a carpel: stigma, style, ovary. In ovary, there is a soft tissue develops known as placenta. On the placenta, a mass of cells differentiates known as nucellus. Now this nucellus is having numerous cells. Out of this, one cell differentiate as megaspore mother cell. It is abbreviated as MMC. Now this megaspore mother cell is diploid. It undergo a process called megasporogenesis. The process in which haploid megaspore develop from megaspore mother cell is known as megasporogenesis. During this process, each megaspore mother cell within the nucellus undergo meiosis I. It gives rise functional haploid cell and the non-functional haploid cell, which will reduce and degenerate. Functional haploid cell undergo second meiotic division. Here it gives functional megaspore and non-functional degenerating cell. So here remember that one megaspore mother cell will develop into only one megaspore. Three cells will degenerate. While in microsporogenesis, microspore mother cell or pollen mother cell undergo meiosis I giving pollen dyad, undergo meiosis II giving pollen tetrad. But here one megaspore mother cell will develop into only one megaspore. Means when you see the ratio in produced microspores and megaspores, it is the 4 microspores and the 1 megaspore. So naturally there is a competition among the microspores. Let us continue. Within the nucellus now, functional megaspore will enlarge. The nucleus of this functional megaspore undergo mitotic division. It is endomitotic division. First mitotic division of a nucleus within the megaspore gives 2 nucleated stage. Second mitotic division gives 4 nucleated stage, and third mitotic division gives 8 nucleated stage. So here within functional megaspore, nucleus undergo 3 mitotic division to form 8 nucleated cell. Out of 8, 4 will occupy micropylar region and remaining 4 will occupy chalazal region. From micropylar and from chalazal region, chalaza is the base, micropylar is the apical region. From each group of 4 nuclei, one nucleus from each pole will move to the center. So, two polar nuclei will occupy the central position. So, now micropylar region will be having 3 nuclei, chalazal region will be having 3 nuclei, and 2 nuclei will occupy the central position. So, 3, 2 and 3. Around each nucleus, the cell wall start developing. So, 3 cells towards micropylar region, 3 cells towards chalazal region will form. So, the middle central cell with 2 polar nuclei is visible. Now, towards micropylar region, the 3 cells are known as egg apparatus, and towards chalazal region, 3 cells are known as antipodals. Here, each nucleus or each cell is haploid. In the central cell, 2 haploid nuclei known as polar nuclei occupying the central position. So now, within functional megaspore, we can see 7 cells, 3 egg apparatus cells, central cell with 2 polar nuclei and 3 antipodals. So 3, 1, 3. Total cells are 7. Now count the nuclei. 1 in each cell of egg apparatus, 2 in the central cell and 1 nucleus in the antipodal. So total 8 nuclei. Earlier what I said, 7 cells and now 8 nuclei. So this is functional megaspore. 7-celled, 8 nucleated structure. Okay, now the position of the megaspore within the ovary during development will change. When we consider functional megaspore with 7 cells and 8 nuclei, and if we take this position, now micropylar region is down, antipodal region is up, central cell in the middle. Okay, now the nucellus cells divide, it forms 2 layers known as integuments. Now sometimes integument is single called monotegument. Sometimes 2 integument layers will form called bitegument. Now these 2 integuments, okay, around the embryo sac or megaspore will leave an aperture known as micropyle. So 2 integuments, micropyle, this is the base called chalaza, the junction where the stalk of the ovule, this is ovule body. Now the ovule is megasporangium. Inside megasporangium there is a megaspore known as embryo sac. Here the stalk of megasporangium is called funicle, right, attached to the placenta. The point of attachment is known as hilum. The base is called chalazal region, towards which antipodal cells we can see 2 integument layers. Here the opening called micropyle. Inside the micropyle, when we see the egg apparatus, the central cell is haploid egg, and either side the cells are called synergids. Now each synergid will be having the flagellar processes known as filiform apparatus. So within egg apparatus, central is the egg, on either side they are called synergids. Like egg is the queen, on either side security guards. Please don't remember these words, just easy to understand. I am giving the example. Egg is the queen of embryo sac waiting for the male gamete, and to protect this female gamete, egg, on either side security guards are standing with the rifle. This is just for explanation I am telling. Okay, these synergids with filiform apparatus. Clear? Okay, now here I took the position of ovary as funicle, the hilum, chalaza, body of the ovule, Micropyle, body covered by two integuments. Inside the integument, soft tissue is the nucellus, nourishing the embryo sac or megaspore, and within megaspore there are 7 cells and 8 nuclei, of which towards micropyle is the opening. Towards that, central one is the egg, on either side synergids with filiform apparatus. Central cell with 2 polar nuclei and 3 antipodals towards chalaza. This we call anatropous ovule, as the funicle and micropyle are side by side. Here, if funicle, hilum, chalaza, ovule body, micropyle. So here, funicle and micropyle, okay, are in the straight line. This we call orthotropous ovule. So depending upon the position, the names are given like campylotropous, amphitropous, anatropous. As per NCERT syllabus, we need to remember anatropous ovule. Okay, let me finish here the structure of carpel. Fine. Each carpel, let us revise at the end. Each carpel has 3 parts. Stigma to receive microspores during pollination, style to carry the pollen tube with generative cell which will give rise to male gametes, and ovary. Inside the ovary, megasporogenesis takes place. During megasporogenesis, what are the events? On the placenta, nucellus, a mass of cells develops. Within that, megaspore mother cell differentiate. This megaspore mother cell undergo meiosis. 3 cells degenerate. 1 cell remain functional. Now this functional megaspore enlarges. Its nucleus undergo 3 mitotic divisions giving 8 nuclei. After arrangement of nuclei towards chalaza, micropyle, center, and after development of the cell wall, we can see the functional megaspore or embryo sac with 7 cells and 8 nuclei. Now this embryo sac or megaspore is protected and nourished by nucellus. Extra protection is given by integuments, maybe single or double. Integuments will leave an aperture towards anterior region known as micropyle. The base of the ovule body is chalaza. The point of attachment is called hilum and the stalk is called funicle. Now the ovary is ready with egg in the egg apparatus guarded by synergids, whose filiform apparatus waiting for the pollen tube who is carrying the male gametes. So entry of the pollen tube is through micropyle. Filiform apparatus are waiting to receive the pollen tube. 2 male gametes will enter, right? And then double fertilization takes place. That we'll study later. Clear? So here for board exam, need to remember female reproductive gynoecium, members are carpels or pistil, structure of carpel with the labeling stigma, style ovary. Within ovary, what events or what are the steps takes place during megasporogenesis which carry 5 marks weightage. Then megaspore or embryo sac. It is 3 or 2 marks question. Okay, they will ask you to sketch and label mature embryo sac or megaspore. For 5 marks with minimum 10 labelings will attract 5 marks for anatropous ovule. So when you see anatropous ovule, it has funicle, hilum, chalaza, integuments, micropyle, nucellus, and 7 cell, the 8 nucleated embryo sac having antipodals, central cell with 2 polar nuclei, egg apparatus having central egg and on either side synergids with filiform apparatus. With this, I will conclude today's session. Hope you follow. Thank you very much.